Hello everyone, welcome to today's tutorial on how to use the SRA Explorer tool to search for SRA data in GOLD using different criteria. This tool provides a window to SRA data and lets you search for SRA data already curated in GOLD or not yet curated in GOLD. Let's get started. To begin, let's navigate to the SRA Explorer tab from the home page. For this demonstration, let's imagine that I'm a microbiologist searching for metagenomes in GOLD, specifically related to the gut metagenome. I'm particularly interested in sequences obtained using the Illumina sequencing platform and focusing on whole genome sequencing. First, let's set the filters. Under the GOLD category, I'll select projects in GOLD. In the scientific name category, I'm going to select gut metagenome under metagenome scientific name. Under library source options, I'll click metagenomic and metatranscriptomic. For sequencing instrument options, I'll select all Illumina instrument options. Finally, for library sequencing strategy options, I will select WGS or whole genome sequencing. Now I will submit my search. Perfect. We have all the search results for our specific SRA search filters. We have 28,613 results. Unlike the other search and refine functions, we cannot directly download the data from the SRA Explorer. However, we can browse through some of the available SRAs to get a better understanding of the sample descriptions and note down the corresponding NCBI biosample IDs for future reference. For example, I will click on the first SRA experiment ID, and here I get the SRA study ID, a study title, sample ID, and the NCBI biosample ID, as well as an NCBI taxonomy ID. Once I get my search results, I can continue to refine the SRA search filters. Let's say, for example, I want only the gut metagenomes from an infant. I can go to text search and type in infant into the SRA study title. This narrows down the search to 3,922 results. And that's it. By using the SRA Explorer, we can filter SRA data based on various criteria to find the specific data sets we are looking for. Remember, you can refine your search using the GOLD, text search, scientific name, library source options, sequencing instrument options, and library sequencing strategy options. Although data download is not available directly within the SRA Explorer, you can browse through the available SRAs to gather more information and make note of the NCBI biosample IDs. Thank you for watching and happy exploring.